1. Introduction. The term, bioindicators, refers to any organisms, species, communities or biological processes, which help to qualitatively monitor the status of the environment in which they live by means of their function as well as their population. The occurrence of any environmental change or problem within an ecosystem can be predicted very easily by any changes in the population status, behavior, and physiology of such organisms. The identification of any species as a bioindicator is reflected by any fluctuation in the abundance of the species in response to any environmental change in a particular habitat. Bioindicators are useful in providing information about the health of an ecosystem since the members of these species are highly sensitive to the changes in their surroundings. Figure 1. Therefore, the sampling and studying about the population dynamics of such organisms makes it possible to monitor any ecological change. This in turn helps in identifying the positive and negative impacts of human activities in the ecosystem. A bioindicator has specific requirements regarding some physical or chemical variables, such that variations in the presence or absence, morphology, physiology, population or behavior of the given species suggest that the given physical or chemical variables are outside their preferred limits. Generally, bioindicators are designated as species which respond to anthropogenic impacts on the environment. An all-inclusive definition of a bioindicator states, a species or a group of species that voluntarily imitates the abiotic or biotic state of an environment, represents the effect of environmental change on a habitation, community or ecosystem or is indicative of the diversity of a subset of taxa or the whole diversity within an area. Bioindicators help in the assessment of the quality of the environment and how it changes over a given period. These can be plants, animals, algae, lichens, zooplanktons, insects, amphipods, mollusks, echinoderms, and other microorganisms. Even human nail and hair can also be used as bioindicators. Bioindicators find usage in monitoring the quality of air, soil and water in any region and in the assessment of the overall biodiversity. These can also help in the determination of the progress of mitigative measures implemented for environmental conservation and thus play a significant role in nature conservation too. The information provided by bioindicators is adequate and reliable and might be difficult to acquire or compute in such a quick manner using other means. Paper 3 Biodiversity and Conservation. The usefulness of bioindicators is most observed in given three situations. When it is impossible to measure the indicated environmental factor. When the measurement of the indicated factor is very difficult. When the measurement of the environmental factor is possible and easy but the interpretation is quite difficult. Figure 1. Response of different bioindicators to environmental stress. Source. Organisms to measure environmental impact 16,821,310. 2. Characteristics of bioindicators. The identification of any organism, species or process as an ideal bioindicator requires possession of certain characteristics such as it should be widely distributed. It should be highly abundant in a certain environment. It should be site-specific and highly sensitive. Paper 3 Biodiversity and Conservation. It should have restricted mobility. It should accumulate and concentrate the toxin to measurable levels. Its position in the tropical system should be clear with fine feeding strategy and a constant rate of metabolism. It should possess a low genetic and ecological variability. Its response should be representative of the responses of the other taxa or the ecosystem as a whole. It should be cost-effective and robust while handling. Its sampling, sorting as well as storage should be simple. It should have an environmental significance and economic importance. Figure 2. Characteristics of bioindicators in the context of their use in biomonitoring. CHARCTERISTICS. A BIOINDICATORS. Specificity. REPRESENTATIVNS. Abundance. Practicability. Social. Importance. Sensitivity. Paper 3 Biodiversity and Conservation. 3. Classification of bioindicators. Bioindicators can be classified into different categories, as illustrated in Figure 3, on the basis of aim of bioindication. Application of bioindicators on the basis of International Union of Biological Sciences, IUBS, Figure 3, schematic diagram for classification of bioindicators. 3.1. Based on aim of bioindication. When classified on the basis of aim of bioindication, bioindicators are of four types. I. Early warning indicators. The bioindicators which can show the first signs of disturbance in the environment are called early warning indicators. These reveal Paper 3 Biodiversity and Conservation. Signs before most other species are affected since they have very quick and sensitive responses to any environmental change. For e.g. ants. E. Compliance indicators. The bioindicators which help in confirming the accomplishment of maintenance or restoration goals are known as compliance indicators. For example, measurement of fish characteristics is a bioindicator for the sustainability of the population or community as a whole. E. Diagnostic indicators. The indicators which help in the examination of the detected environmental changes or disturbances are known as diagnostic indicators. These indicators are generally measured on the individual or suborganismal level. IV. Accumulation indicators. The bioindicators which help in studying the effects on different biological organization levels are known as accumulation indicators. For e.g. mosses, lichens, etc. 3.2. Based on different applications of bioindicators. When classified on the basis of applications, bioindicators are of four types. I. Environmental bioindicators. These refer to the species which respond to any disturbance in the environment and thus reveal the signs of any environmental change or disturbance. They help in the diagnosis of the environmental status for making any environmental policy. For e.g. animals, macroinvertebrates, sentinels, etc. E. Ecological bioindicators. The species which are sensitive to fragmentation of the habitat, pollution or other disturbances or stresses in the environment are called ecological indicators. For e.g. lichens, plant indicators, etc. E. Biodiversity bioindicator. The species which are the indicators for measurable parameters of biodiversity such as genetic parameters, landscape parameters, etc. and represent the species richness of a community are known as biodiversity bioindicators. For e.g. animals, plants and microbial indicators. Paper 3 Biodiversity and Conservation. IV. Pollution bioindicators. The bioindicators which are helpful in the detection of presence of pollutants in a region are known as pollution bioindicators. For example, animal and plant indicators. 3.3. Classification of bioindicators on the basis of International Union of Biological Sciences IUBS. The International Union of Biological Sciences IUBS has divided bioindicators into following 6 groups after recognizing the importance of bioindicators in monitoring environmental changes. Microbiology, botany, zoology, cell biology and genetics, comparative physiology, hydrobiology. 3.3.1 Microbiology. Microbes quickly detect different types of the environmental pollution such as soil and water pollution. Some microbes help in the decomposition of some pollutants while some of them are sensitive to some toxins or harmful substances. The reduction in the population of the sensitive species or increases in the population status of the tolerant species indicate changes taking place in the environment. These changes in the species diversity may be the result of the presence of some toxic substances. For example, presence of sulfur can be easily detected in the microbial muds from continental and intercontinental water bodies. Some microbes are also helpful in the assessment of variations induced in the marine environment by various anthropogenic activities such as E. coli, Pseudomonas, Clostridium, Streptococcus, etc. The presence of pesticides in soil is indicated by cyanobacteria. For example, nostoc microscopicums indicates the pollution of soil by pesticides such as dithane, delton, ferrate, etc. Paper 3 Biodiversity and Conservation. Oil spillage or oil pollution can be easily monitored using yeast, actinomycetes, some filamentous fungi, and bacteria. Some microbial species such as Penicillium sps and Oreobasidium pullulans can utilize oil fractions from waste oil. 3.3.2. Botany. This group can further be subclassified into the following 2 categories since different groups of plants indicate the changes in the environmental status. Lower plants, higher plants. A. Lower plants. The taxa involved in lower plants as bioindicators include a wide range of lichens, algae, fungi, leaf yeasts, etc., which help in the detection of both short-term as well as long-term effects on the basis of appropriate choice of organisms. The tolerance level or resistance towards a substance in an environment varies with species. For example, lichens are considered ideal bioindicators or monitoring agents due to their vulnerability and resilience to different environmental effects. Some lichens can bloom only in the fresh or unpolluted air while some of their species are resistant even to much polluted air. The lichen lecanora is good biological indicators of a wide spectrum of the environment. The lichen thalli indicate the presence of sulfur dioxide and fluorine in the atmosphere. Lichens are also used for survey of long life. The presence of some radionuclides such as cesium-137, strontium-30 released during nuclear explosions in the environment can also be monitored using lichens. The lichen thalli are able to absorb fluorine and heavy metals including lead even when they are dead. Lichens generally thrive on rocks and tree surfaces or soil and do not have roots and cuticle. They attain all their nutrients directly from the atmosphere rather than from the soil. They are such ideal monitoring agents because of their extreme sensitivity to the toxins present in the air as well as due to their high surface area to volume ratio which further leads to the accumulation of toxins or contaminants from the air. Figure 4. The lichens are also Paper 3 Biodiversity and Conservation. Considered because of being cost-effective and inexpensive agents in the evaluation of air pollution when compared to most of the physical and chemical monitors. Figure 4. Lichens as bioindicators. Source. Various algae are too used in environmental monitoring such as Viva and Enteromorpha for monitoring the water quality of estuaries, Cladophora and Stagioolonium for heavy metal contamination of water, Chlorella for monitoring the presence of toxic substances in water bodies, Skeletone Macostatum, Amphidon carterae, Pavlova lutheri, etc. For the indication of oil pollution, etc. B. Higher plants. A wide range of higher plants serve as bioindicators. Air pollution is monitored using some sensitive species whereas some tolerant indicator species are used for the determination of soil conditions. Plants also serve as indicators of presence of various heavy metals such as Al, Ca, Co, Cu, Zn, Cd, Pb, Hg, Mo, Mn, Ag, etc. And have also been used to detect and monitor gaseous pollutants such as sulfur dioxide, SO2, ozone, O3, nitrogen oxides, etc. Figure 5. Paper 3 Biodiversity and Conservation. Figure 5. Ozone damage to plants. Source. Some of the well-known examples of higher plants as bioindicators as well as pollutant scavengers include Anthroxanthum spp is tolerant to zinc. Agrost is tolerant to copper. Festuca is tolerant to lead. Odiolempideme is tolerant to calcium. The plants serve as ideal monitoring agents due to various phenotypic, metabolic and anatomical changes in the plant system and the type of changes help in the determination of the nature of compounds to which the plants are exposed. The damage caused on exposure to ozone gas can be reflected by the weathered flakes of tobacco or chlorotic flakes of pine needle. The damage caused by peroxyacetyl nitrate can be indicated by the collapse, glazing and bronzing of leaf cells in plants. Some of the physiological and anatomical changes in plants include the effects on the nature of stomata, pigmentation, chlorosis and bleaching. For example, the inhibition of Paper 3 Biodiversity and Conservation. Photosynthesis and enolase due to fluorine damage, bleaching of perianth and stamen injury by mercury poisoning, etc. 3.3.3. Zoology. Valuable data about the accumulation of chemicals in animals and their role as efficient biomonitors can be achieved by the study of both individual species as well as whole communities. Accumulation in animals occurs in different degrees in different organs, which allow more effective choices for choosing suitable indicator species. The animals are designated as bioindicators even when there is a relative absence of accumulation, since marked changes have been seen to be induced in the development and reproduction of particular species on being subjected to environmental pollution. The bioindication in animals also takes place by the accumulation of chemicals within food chains, which further leads to biomagnification. Some of the examples of animals as biological indicators include heavy metal and pesticide pollution in water can be monitored by fish, daphnia, silver carp, etc. Fresh waters can be indicated by the presence of zooplankton such as rotifers and cladocerans. Radioactive pollution in the soil can be indicated using earthworms. Frogs. The health of frogs is an indicator of the health of the biosphere as a whole because most frogs generally require suitable habitat in both the terrestrial and aquatic environments. The frogs are generally very vulnerable to environmental changes and disturbances since their skin absorbs toxic chemicals very easily, and thus they make ideal indicators of environmental pollution. 3.3.4. Human system as bioindicators. Human cells, tissues, organs, organ systems, etc. also act as effective indicators of environmental stress. Human blood and urine can indicate the presence of toxic compounds. Paper 3. Biodiversity and conservation. On exposure to such substances. For instance, human hair can trap metallic vapors and dust over a long period of time due to the affinity of these substances with the hair protein. 3.3.5. Cellular and subcellular components. The degree of tolerance, vulnerability, and compliance in living organisms to various environmental factors and states are controlled by their characteristic genetic composition and physiological states. The process of biomonitoring can be effectively carried out by the examination of cellular and subcellular components of the living organisms, including chromosomes. Any chromosomal or genetic damage precisely indicates the extent of certain mutagenic agents in the environment. In resistant organisms, which are tolerant to toxic substances, a degradation of the pollutant may take place, and thus they make excellent indicators of environmental stress. 3.3.6. Comparative physiology. The sense organs and other organ systems in various animals show behavioral responses on exposure to any kind of environmental stress or disturbance. Exposure of an organism to a pollutant may lead to coughing, interference with vital ionic or gaseous changes in skin pores, etc. A chemical may affect the functioning of various systems such as endocrine, nervous, muscular, cardiovascular, and excretory systems. Such changes may be examined at morphological, biochemical, or physiological levels in an organism and can thus indicate the presence of toxic substances. 3.3.7. Hydrobiology. Macrobenthos as well as other biota living in the aquatic systems can be used to indicate the quality of various water systems. Biotic index in any water body is one such parameter for the purpose of biomonitoring. 4. Relevance of bioindicators. The biological relevance of a bioindicator species is determined by certain characteristics such as little natural variability, stimuli to environmental stress, specificity, sensitivity, and Paper 3. Biodiversity and conservation. The acquisition of biologically valid as well as measurable changes. An early warning indicator of potential adverse effects of any disturbance is the most useful indicator. However, a very sensitive response may lead to a false indication of very minor or biologically invalid changes and thus may not be considered relevant. An ideal biological indicator is the one reflecting irreversible damage as well as measurable changes. 5. Importance of bioindicators. 5.1. Ecological importance. Bioindicators help in the determination of environmental changes, monitoring the level of pollution and its effects, and the health of an ecosystem as a whole. Bioindicators of different types provide numerous applications, for instance, the indication of increasing eutrophication and organic loading on coral reefs, especially sponges. Bleached and broken corals and coral mortality are the indicators of past stress on coral reefs. 5.2. Assessing human and ecological health. Ecological health includes the aspects of human health as well as the climatic conditions and water quality along with food stability for survival and aesthetic values. Ecological health involves proper maintenance of species diversity and populations as well as that of the functional characteristics like competitive interactions of the ecosystem. The sustainable availability of goods and services to all organisms in an ecosystem is an important aspect of a healthy ecosystem. Bioindicators are frequently used for the assessment of ecological health. 5.3. Monitoring water quality. Bioindicators help in efficient and cost-effective determination of water quality. For instance, the cellular metabolism of bioluminescent bacteria gets inhibited by the toxins present in water, which leads to an effect on the intensity of light emitted by the bacteria. Thus, they are suitable for the analysis of water quality for the presence of environmental toxins. Water quality can also be monitored by the study of zooplankton community in aquatic systems and using macroinvertebrates such as bugs, since a decrease in their population status reflects degradation of water quality. Some tolerant bioindicator species include annelid worms and Paper 3. Biodiversity and conservation. Red midge larvae which are capable of breathing at the water surface and thus can thrive in polluted waters. Thus, different species respond in a specific manner to various environmental disturbances and can be used as efficient monitoring agents. 5.4. Monitoring air quality. Bioindicators can be used effectively for the assessment of air pollution, which provides an early warning of sites which are vulnerable to the deposition of air pollutants. Some of the methods serving this purpose are epiphytic macro-lichen frequency, chemical analysis of mosses and community composition, etc. Lichens and mosses can be used to monitor the quality of air by the measurement of various air pollutants such as fluoride and chloride content, ozone content, heavy metals, presence of radionuclides, etc. Therefore, lichens are considered as the high-quality indicators to monitor air pollution. 5.5. Monitoring biodiversity. The current biodiversity crisis has led to an immediate need for urgent conservation policies and the identification of significant sites that promise the accomplishment of conservation goals. Bioindicators are potentially beneficial tools in achieving this. The disappearance of some noticeable animals such as birds, butterflies and mammals can play a major role in the monitoring of forest ecosystems. The usually faced problems in the process of biodiversity conservation involves the absence of adequate resources such as sufficient data, and these can be improved by conservation of some taxa keystone species, which in turn provides the advantage of the protection of other taxa, species, or the complete biodiversity. Thus, indicator species can be effectively used as a conservation tool to recognize biodiversity hotspots. 6. Applications of bioindicators. Bioindicators have a wide range of applications. Some of these are as follows. Paper 3: Biodiversity and Conservation. Bioindicators can be effectively used as the indicators of water quality and thus, water pollution. For e.g., the presence of E. coli in water bodies is the indicator of water polluted by fecal matter. Bioindicators can be used for the assessment of air pollution. For e.g., the lichen thalli indicates the presence of fluorine in the atmosphere. Bioindicators determine an environmental disturbance or stress very easily and accurately. Bioindicators help in the assessment of the quality of the environment and thus play an important role in the maintenance of health of an ecosystem. Eutrophication in water bodies is indicated by the bioindicators. For e.g., presence of algal blooms in water body is the indicator of nitrates and phosphates. Bioindicators help in the measurement of the extent to which soil has been polluted. For e.g., cyanobacteria are used as bioindicators of soil pesticides. Bioindicators can be effectively used as a conservation tool to recognize biodiversity hotspots. 7. Advantages of bioindicators. Bioindicators are relevant, cost-effective and very reliable agents for environmental monitoring since the effect of pollutants on indicator species is clearly recognized. The techniques involved in bioindicator-based studies are relatively simpler, repetitive and feasible in a number of ecosystems. Indicator species are significant to monitor the environment because of their easy interpretation and less ambiguity than direct sampling and assessment of all plant and animal communities found in a certain ecosystem. 8. Disadvantages of bioindicators. Environmental monitoring using bioindicators experiences a number of limitations such as the presence of sufficient number of individuals of the indicator species at the given locality, abundance and wide availability of species in the area of interest along with sufficient information about the physiological processes of uptake and accumulation of toxic substances or environmental contaminants in the indicators. Paper 3: Biodiversity and Conservation. Conclusion. Bioindicators are biological tools which help in the determination of various disturbances taking place in the ambient environment. Several plants and organisms such as microorganisms, algae, lichens, mosses, invertebrates and vertebrates are used as bioindicators to analyze changes in the environment. Bioindicator-based studies are usually simple and repeatable at different times by different individuals in different environmental conditions and are also suitable for the assessment of large areas. Bioindicator species indicate contaminant exposure and toxicity mechanisms in the environment, provide early warning of future environmental damage and early indications of environmental recovery, thus help in ecosystem conservation. 5. Paper 03: Biodiversity and Conservation. 3. Description 3.1 Introduction. The term biopiracy is oftentimes used as a synonym for bioprospecting, however, there are subtle differences. The differences are indeed a question of politics and ethics. While bioprospecting implies that the samples are acquired through proper legal and ethical frameworks, including equitable benefit sharing and prior informed consents, the term biopiracy is used when an indigenous knowledge is patented for lucrative purposes with neither the knowledge of indigenous community nor their consent. However, the difference is so subtle that it is oftentimes an ethical and political question. Political philosophies that are against globalization, for example, socialist and left-wing ideologies, oppose any form of bioprospecting and blame it as biopiracy. For example, biopiracy is oftentimes defined as a practice commercially exploiting naturally occurring genetic material or biochemical. Even though a consent is obtained from the chieftain of an indigenous tribe to obtain the sample, how ethical it is to take the ownership and patent it. However, there is a consensus that biopiracy is exploitation of marginalized or tribal or indigenous communities. Bioprospecting might or might not involve such communities. For example, as discussed in Bioprospecting module, when a Swiss scientist developed a blockbuster drug from microbes isolated from soil samples that he collected from Norway on a family holiday, the case had nothing to do with indigenous community and is not regarded as a case of biopiracy. Biopiracy is generally regarded as unethical, in contrast with bioprospecting, which is an ethical practice. At the heart of this issue lies the concept of ownership. Can a firm or community or a nation claim ownership for a particular species of organism? Similar situation can also be found in the discipline of taxonomy. A number of plants extensively used in indigenous medicine, for instance Tulsi Ocimum tenuiflorum, had been formally described by Swedish taxonomist Carl Linnaeus, and he is the taxonomic authority of 6. Paper 03: Biodiversity and Conservation. These plants. It is not the case that these plants have had no names in Sanskrit. Tulsi is the Sanskrit word for this plant, yet the credit for naming this plant had been taken by the Swedish scientist. Patenting rules differ from country to country. In India, patenting a plant species or any living organism is not allowed, while the U.S. Patent Office grants such patents. As India do not come under the purview of U.S. Patent Office, on first glance this issue seems irrelevant. However, oftentimes these plants could be developed into transgenic varieties and become a commodity of multinational monopolies, three most prominent firms being Monsanto, DuPont and Syngenta. Oftentimes, the firms make these transgenic plants deliberately infertile such that the farmers need to purchase the plants again and again from the companies. The reason why bioprospecting firms often target indigenous knowledge is obvious. This traditional knowledge with their trial and error approach, albeit the approach itself is unethical and cumbersome, might offer better prospects for successful lead discovery rather than random screening of natural products. Random screening is especially cumbersome. It is estimated that to develop one successful drug candidate, the company need to screen a very large number of samples, approximately 250,000 of it, and is therefore extremely slow, takes decades to develop a drug, and is very expensive. Oftentimes, even after screening, the firm would come out with a drug for which better and cheaper substitutes are already available in the market, rendering the lead development effectively useless. As the bioprospectors largely hail from rich countries of temperate regions and they access samples from poorer countries of tropics, the question on how sample was accessed oftentimes becomes a political question. Analogy is how ethical the British government possessing Kohinoor diamond that they accessed during the colonial times, or numerous such cases, accessing ancient Egyptian mummy specimen for instance. One such a case is discussed later on that involves sample acquisition by a 7. Paper 03 Biodiversity and Conservation. French firm from a former French colony in Africa. Biopiracy is, therefore, oftentimes linked with colonialism. Apart from acquisition of plant or organic materials, such as snake venom, as discussed later, biopiracy also covers genetic resources. Genes of wild races are involved with various desirable attributes, such as disease resistance, draft resistance, tolerance to heat and salt stresses, high yield and so on. These genes obtained from the nature can be cloned and developed into transgenic organisms possessing such desirable qualities. Genes coding for desirable proteins can be cloned and expressed in bacteria for the direct production of such substances in bioreactors, for example, production of insulin and Taq polymerase. Therefore, if such genes are obtained from sample materials that are used as part of indigenous medicinal systems without proper ethical and legal frameworks, that would construe as a case of biopiracy as well. 3.2 Famous case studies of biopiracy Maya ICGB. This controversy happened in 1999-2000 when two scientists from University of Georgia, USA established International Corporative Biodiversity Group, ICGB, to document the biodiversity of Chiapas region of Mexico and ethnomedicinal features of indigenous Maya people. ICBG established an NGO ProMaya in Mexico as a mediator. The controversy was that PROMAYA was wholly under the control of two U.S. researchers and benefit sharing and PIC merely remained in the paper, a clear case of unethical exploitation of indigenous knowledge, or a case of biopiracy. The project was terminated in 2001 without any bioprospecting success. However, this case was the first to highlight the perils of biopiracy. 8. Paper 03 Biodiversity and Conservation. Basmati rice. Basmati rice, an indigenous long-grain rice native to India, is one of the major exporting commodity of India to US, Middle East and EU markets. Rice Tech, an American firm, filed for a patent for certain hybrid of basmati rice that they claimed developed by the firm in 2000, and filed for the exclusive use of their trademark basmati in US markets. After the intervention from Indian government, several claims of the patent were invalidated. As basmati rice is a registered geographical indication, patenting on this or its hybrids is being protected by the European Commission. Darjeeling tea. Darjeeling tea is a tea variety cultivated at Darjeeling, West Bengal for the last 2 centuries, and is generally regarded as a unique commodity produced by India. Of course, India is the world's largest producer of tea, with a global share of about 35%, and Darjeeling tea exports generate a substantial revenue for the tea farmers and industries in India. However, in a number of developed countries, especially Japan and France, the word Darjeeling is awarded a trademark to their own private firms, although the product itself might not be from the Darjeeling region of India. This situation is in stark contrast with other GI producers like champagne, France, tequila, Mexico, shiitake mushroom, Japan, and so on. Quassia. Quassia amara is an indigenous plant that grows in French Guiana, the former French colony in Africa. A French institute successfully developed an anti-malarial compound from this plant and received an EU patent in 2015. However, extracts of this plant was in use for many centuries as part of the indigenous medicinal systems of French Guiana. The French patent applicants claimed that their extract is alcohol-based, while that of the traditional medicine was water-based. Indeed, this is a valid point for uniqueness and novelty, 2 criterions that the Patent Office check before granting the 9. Paper 03 Biodiversity and Conservation. Patent. Alcohol-based extracts usually isolate different set of compounds from that obtained from water-based extracts. However, no prior informed consent was obtained by the French Institute before obtaining the plant sample. The case is still sub judice as of this writing. Neem. W.R. Grace, an American firm along with U.S. Department of Agriculture, obtained an EU patent in 1994 for antifungal chemicals that they have extracted from neem trees, Azadirachta indica. However, neem is being used for centuries in Indian traditional system of Ayurveda. Prominent environmentalist groups from around the world, including Vandana Shiva from India, opposed this patent in 2000. Finally, the EU Patent Office revoked the patent in 2005. Turmeric. At least 4 U.S. patents were granted on the medicinal uses of Indian plant turmeric. The famous among them is the patent filed by 2 Indian expatriate scientists from University of Mississippi Medical Center, U.S. for the use of turmeric powder in wound healing. India's CSIR filed an application with the U.S. Patent Office to revoke the patent as the turmeric had been in use in Ayurveda for thousands of years. The CSIR even produced ancient Sanskrit texts and previous papers on the medicinal uses of turmeric powder. In response, the U.S. Patent Office revoked not only this particular patent, but also all the rest 3 patents for the uses of turmeric. Tamarind. World's largest producer of tamarind is India, with annual production more than half of the world's total. Tamarind pulp had been part of the traditional Indian Ayurvedic medicine for last many centuries as a component of various herbal formulations for treating various diseases. From 1978 until 1997, there were about 30. 10. Paper 03 Biodiversity and Conservation. U.S. and Japanese patents each granted for the tamarind-based pharmaceutical formulations. At the same time, Indian organizations like ICAR and CSIR owns merely 8 patents in India for the applications of tamarind paste. Having granted more than 60 patents worldwide on tamarind paste, which Indians consider their traditional intellectual rights, this case have received a lot of media attention in India to such an extent to call all those international patents as a case of biopiracy. Okra. In 1999, U.S. firm Chromic Research Inc. obtained a U.S. patent for an herbal mixture that contained extracts of okra Kerala bitter gourd. However, okra juice had long been used in indigenous medicinal system of Ayurveda for treatment against diabetes. This patent received a lot of media fury in India and was termed yet another case of biopiracy. Jar Amla. Fox Chase Cancer Center in Philadelphia, USA was successfully awarded an EU patent for the extracts from jar amla plant Philanthus neruri for treatment against hepatitis B. However, this plant had been used for centuries in Indian traditional medical systems including Ayurveda, Siddha and Unani for treating jaundice. Brazine berries. In University of Wisconsin at Madison, U.S. obtained 3 patents for the brazine berries Pentadiplandra brazine, including one for a low-calorie artificial sweetener obtained from this plant which is 2,000 times more sweet than the ordinary. 11. Paper O3 Biodiversity and Conservation. Cane sugar. The plant is indigenous to West African country of Gabon and its qualities were known for the West Africans for centuries. It was said that the researcher noticed the Gabon people eat the berries and thought that there might be something special about the berries to investigate its potentials. The UW-Madison team isolated the gene from the plant coding for the protein, and they cloned it in the suitable vector to commercially harvest the sweetener at large quantities without having to go to Africa to collect the plant or to grow it there, and received yet another patent for this. The turnover of U.S. artificial sweetener market is estimated to be around $100 billion United States dollars, yet the UW-Madison shared none of the generated benefits with Africans. Captopril. The drug captopril belongs to a family of drugs called angiotensin-converting enzyme ACE inhibitor used to treat hypertension and congestive heart failure. Captopril was developed from the venom of lance head viper snakes Bothrops jararaca that are native to Amazonian rainforest. However, before the development of this drug, venom of this snake had been used by the indigenous Brazilian tribes to coat it on the arrowheads used in hunting and intertribal warfare. The drug was developed by the American firm Bristol-Myers Squibb and patented in 1977, however no profit sharing was made by them to the Brazilian tribes. Enola bean. The Mexican yellow bean is an edible bean cultivated throughout Mexico. In 1999 a patent for this bean was granted by the U.S. Patent Office. The bean was named Enola in the patent application after the name of the wife of the patent applicant, Enola. The patent holder sued a large number of Mexican exporters who exported these beans. 12. Paper O3 Biodiversity and Conservation. To the U.S. citing the patent law infringement. The impacts were truly tremendous. Over 90% of sales dropped negatively affecting more than 20,000 farmers. Mexican farmers filed an application with the U.S. Patent Office to revoke the patent. In 2008, the U.S. Patent Office ruled in favor of Mexican farmers and revoked the patent. Quinoa. In 1994, University of Colorado, U.S. obtained a broad-spectrum patent for the plant quinoa Quinopodium quinoa, which is a staple food for many tribal populations living in Andes ranges, South America. Quinoa is the principal food source for Quechua tribes of Peru, Chile and Bolivia and they pressurized the university to abandon the patent in their interest. As the Bolivia exports quinoa to the tune of $1 million United States dollars per year, the patent would affect the whole export, and finally to the farmers largely. Finally, the University of Colorado abandoned this patent. Rosy periwinkle. Rosy periwinkle Catharanthus roseus is a native poisonous plant from Madagascar. However, during the days of colonialization, this plant was introduced at many different countries throughout the world. Its traditional African use was against diabetes. Various tropical countries where this plant was introduced claimed that the plant had been part of their indigenous system for the treatment of numerous diseases, including sore throat, pleurisy, dysentery and so on. At least 2 potent and commercially successful chemotherapeutics against cancer were developed from this plant, viz. Vinblastine and vincristine, although no benefits were shared by the American drug firm Eli Lilly either with Madagascar or with other tropical countries where this plant is used as part of their traditional medicine. 13. Paper O3 Biodiversity and Conservation. Bt brinjal. U.S. agricultural firm Monsanto, through its Indian subsidiary May Company, obtained the seeds of Indian varieties of vegetable brinjal aubergine eggplant without prior informed consent from National Biodiversity Authority (NBA) of India. The firm developed insect-resistant transgenic varieties of brinjal by cloning cry-protein genes from Bacillus thuringiensis that express a potent toxin delta-enterotoxin specifically targeted to the insects while harmless to humans. The firms, in collaboration with local agricultural universities, undertook large-scale field trials in Tamil Nadu and Karnataka in 2005-06. However, there was no permission obtained from NBA before conducting field trials as well, an obvious case of biopiracy. Subsequently, NBA initiated judicial trials against Monsanto. However, it turned out that the firm had already obtained permission from India's Department of Biotechnology in 2005. This case reveals administrative inadequacy and lack of effective interdepartmental communication. Artemisinin. Extracts of the wormwood plant Artemisia annua had been part of the traditional Chinese medicine for more than 2 millennia for treating malaria. In 1979, Chinese researcher Tu Yu isolated the compound artemisinin from the plant extracts and immediately published the findings in Chinese medical journal instead of patenting it. Since then, the artemisinin and its other synthetic derivatives have become a standard drug for the treatment against malaria and saved millions of life worldwide. As the drug was not patented, no drug firm held monopoly and generic medicines were readily and immediately available worldwide, albeit patenting could have earned China billions of dollars in revenue. This case illustrates a recurring ethical theme involved. 14. Paper O3 Biodiversity and Conservation. With life-saving drugs, is patenting ethical? However, most of the drug screening and bioactivity investigations for the drug development are being undertaken by private R&D firms, and for their sustenance, profit generation via patenting is required. Tu Yu won Nobel Prize in 2015 for this discovery. Hoodia. Hoodia gordoni is a leafless succulent plant native to Kalahari Desert in South Africa, and grows abundantly in Botswana, Namibia and South Africa. Indigenous tribes, San people, used extracts of this plant as a panacea, magical medicine against various ailments, and as an appetite suppressor. Samples of this plant were acquired by American multinational firm Unilever, and they signed MO with South African Council for Scientific and Industrial Research to develop a dietary supplement. While San people were not part of this deal, later on they were invited and made part of the benefit share. After a decade of research and spending nearly 20 million United States dollars, Unilever finally abandoned the program because of the toxicity of the plant and negative results. This example illustrates problems with bioprospecting in general. Success rate is extremely low indeed. 3.3 Current slowdown. The case of Hoodia just discussed highlight a major issue with bioprospecting. It is immensely a slow process. There is a large scope for potential controversies, extremely expensive, and chances of success very low. Post-implementation of CBD and Nagoya protocols, sample acquisition from third world tropical countries have become a major bureaucratic huddle for bioprospecting firms from rich temperate countries. In the last 2 decades or so, a far better and straightforward approach of 15. Paper O3 Biodiversity and Conservation. Combinatorial chemistry have started emerging. Combinatorial chemistry aims at synthesizing key ligands that specifically target certain molecules of a disease pathway. Instead of screening a large number of natural products, this approach first look inward, the disease itself. Structures of key molecules involved in disease progression are generated, and ligands that best bind with those molecules are chemically synthesized. This approach is far more specific and directional, unlike trial and error approaches used in screening of natural compounds. Yet another approach is, instead of screening the natural products, a number of pure compounds are isolated from each extract, these pure compounds subjected to structure elucidation, and the resulting structures housed in a public database. Researchers would now be able to dock structures of potential target molecules of disease pathways with that from the database, an in silico approach known as molecular docking.